Hey, Jiajia, I recently grew a painful ulcer, and my mom said that taking herbal tea will help. Guess what? There's actually some scientific backing behind it.
Oh, what did you find?
Well, our oral cavity acts as an important bridge between our body and the environment. You know that different parts of our mouth contain different microbiota, and these microbiota are influenced by both our body's natural factors and the environment. There's actually a growing body of research that links specific oral bacteria to certain health conditions.
So if the balance of bacteria gets thrown off, it could not only lead to local diseases like ulcers and gum disease, but also systemic diseases.
Yeah, for example, periodontal disease is due to the dysbiosis of the oral microbiota guided by inflammatophilic bacteria, leading to an altered resolution of inflammation and lack of regulation of the inflammatory response. To counter this, the usual approach is to use antibiotics to fight the infection. However, with the rising problem of antibiotic resistance, people are looking into other options, something natural. However, it could be due to the systemic disease leading to changes in the oral microbiome as well.
Oh, that makes sense. So what other alternatives are we talking about?
Well, after much research, alternatives like diet changes helped, especially for plant-based stuff like herbal teas. Turns out that tea has anti-inflammatory and antioxidant properties that might potentially benefit the oral microbiome.
Wait, tea? Like my morning cup of tea could be good for my health?
Potentially, yes. Here, a cross-sectional study has shown that tea drinkers have a richer and more diverse oral microbiome compared to non-tea drinkers.
Wow, that's something I didn't expect. I guess with the rise in tea consumption worldwide, it's worth paying more attention to how these beverages could be a natural way to manage oral health.
Exactly. In fact, global tea production and consumption has both been on the rise. Understanding how herbal teas affect our oral microbiota could reduce reliance on antibiotics for managing oral and systemic diseases.
Cool. I never thought they'd have anything to do with oral health.
Definitely. You can see that there's growing interest in studying how regular consumption of herbal beverages, especially those made from medicinal plants, could unlock new strategies for promoting oral health naturally.
Sounds like I need to up my tea game.
Right? The more we understand about these natural remedies, the better we might be able to manage oral health sustainably. How about we start the investigation right now?
Right now.
Yeah, here's a can of Jiajia herbal tea with no sugar.
Zero sugar?
Yeah, since sucrose has been shown to alter both oral and gut microbiome, it's best that we eliminate sugar. Rather than go for herbal breweries, we can ensure consistency through commercial products like this.
Oh, but no sugar means there must be artificial sweeteners, right? Wouldn't that also alter the oral microbiome?
Yeah, this version uses sucralose, an artificial sweetener in place of sucrose. While sucralose is known to alter the gut microbiome, there's yet to be any consensus in its effect on the gut microbiome in typical consumption dosages. Besides, by looking at the oral microbiome, effects of sucralose on the oral microbiome are largely unknown and are limited to reduction of biofilm formation caused by Streptococcus species. I think this is our best bet.
Agreed. Jiajia does have a well-defined ingredient list. Their website lists 11 premium herbs, some of which have been shown to confer health benefits. For example, the Grosvenor myrmidika fruit, also known as luohanguo or monk's herb, has been shown to have antioxidant, antimicrobial, and immunomodulatory effects.
That's right. The drink also contains the spreading hilly of this herb, which has been shown to have anti-inflammatory and immunomodulatory effects.
Cool. These herbal plants are typically used in TCM. There's growing attention on them since experiments have shown that they have a real measurable impact on our bodies.
Yeah, if taking Jiajia does alter our oral microbiome for the better, not only will this support existing research on the physiological effects of herbs, it can also guide subsequent determination of Jiajia specific constituents that play a role in modulating the oral microbiome. But, as a scientist, we should not just accept what people has claimed in their research. Let's continue planning our experiment.
Yeah, how should we go about this experiment?
I'm thinking we need to have a 7-day washout period before we start our 7-day intervention.
Oh, that's so long, a 7-day washout period. Why do we even need a washout period?
The aim is to reduce confounding dietary factors that have been shown to alter the oral microbiome, such as alcohol, fermented food, drinks, probiotics and prebiotics, medication, and lastly, from different types of tea. We should also refrain from eating raw meat, as it will introduce additional bacteria to our mouth. And we need 7 days to give enough time for our oral microbiome composition to reset to its original form, meaning the composition that will be achieved without adding dietary factors that alter the oral microbiome.
Oh, I get it. And the reason behind the 7-day intervention is because we need to give the Jiajia tea enough time to alter our oral microbiome, right?
Exactly. Following existing protocols for microbiome studies, there will be a 7-day washout followed by a 7-day intervention. We need to drink 1 can of 300 ml Jiajia zero sugar within 30 minutes of each day of the intervention between 12 to 2 p.m. without using a straw. A straw can prevent the Jiajia tea from spreading out to our oral cavity, thus preventing it from influencing our oral microbiota.
But is 1 can of herbal tea a day enough?
Well, based on the recommended intake, they recommend us to take 1 to 2 cups a day. Generally, people also take 1 to 2 cups of tea per day, so 1 can is enough to imitate the real life situation.
Oh, that sounds interesting. I can't wait to start an experiment.
Right. So this will be an exploratory investigation on the effects of short-term consumption of herbal beverages. Because of the antioxidant, antimicrobial, and immunomodulatory effects of Jiajia specific ingredients, I would think that this will make the oral mucosa a more habitable place for commercials to flourish. I will hypothesize that the short-term consumption of the herbal tea will increase oral microbiome diversity.
I agree. Findings from this study could guide subsequent investigation into the determination of active ingredients within herbal beverage formulations that promote a healthy microbiome composition, enabling the identification of novel and specific therapeutic agents against oral diseases.
Then how and when should we collect our samples?
We can collect our baseline samples between 12 and 2 p.m. on the last day of the washout period, and our post-intervention samples also on 12 to 2 p.m. on the last day of our intervention period. But don't drink right before we swab, as drinking can wash out some of the microbes.
Okay, but is there any specific region of the mouth that we should swab?
We need a representative sample of the overall oral microbiome, so we'll need to swab different parts of the mouth. I think we should do the upper palate, teeth, right and left buccal mucosa, and lastly the tongue for about 5 seconds while rotating the swab, and use a different swab for each area.
I see. Then we can reconstitute them into one sample.
There are some strengths and limitations for our sample collection protocol. Firstly, an advantage is that compared to just saliva collection, our method increases the likelihood of us obtaining bacteria that also adhere to the mucosal surfaces, increasing bacterial yield. However, because subjects collect their own specimens, swabbing techniques may vary slightly between individuals. Inconsistency in pressure and thoroughness may affect concentration and composition of bacteria collected. Furthermore, swabbing alone recovers approximately 15 to 64% of DNA, so the quantity of bacterial DNA will vary between specimens. Finally, because different bacteria occupy different regional niches within the oral cavity, pulling regional samples would mean that the results are only representative of the whole oral cavity microbiome, rather than being specific to each region. Other than standardizing our sample collection methods, we also realize that as different individuals, all of us have different lifestyles and habits which may influence the types of bacteria that inhabit our oral cavity. Thus, as these factors may affect our findings but cannot be controlled within the design of our experiment, we decided to collect key information which may significantly affect our data collection and analysis. These include our oral hygiene practices, key lifestyle choices which may affect the oral microbiome, and our dietary habits which are known to largely impact the composition of the oral microbiome. Oral hygiene practices that we recorded include the use of antiseptic mouthwashes, dental floss, tongue scrapers, dental appliances like braces and retainers, as well as the frequency of toothbrushing and duration of mouthwash gargling. These factors, especially the use of mouthwashes containing chlorhexidine, are found to decrease bacterial diversity and may inhibit health-associated oral bacteria including Veillonella and Actinomyces. Also, smaller differences in oral microbiome post-intervention may be observed in individuals with stricter oral hygiene routines. Next, key lifestyle factors like cigarette smoking, vaping, and antibiotic use were also recorded. Smoking has been shown to alter the oral microbiome in both American and Chinese populations, and the tongue may contain more pathogenic bacteria among smokers. Antibiotic use has also an obvious impact on the composition of the oral microbiome, and thus these factors were recorded to enhance the quality of our data analysis. Lastly, dietary habits like vegetarianism, consumption of sugary drinks, alcohol, probiotics and prebiotics, and tea and coffee have been shown to alter oral microbiome. Hence, we collected the frequencies of consuming these aforementioned foods prior to the 7-day washout period.
I think there are some limitations to acknowledge. Can you all think of any?
Oh, varying diets among subjects might affect the oral microbiome so that the differences observed pre and post intervention are not purely due to drinking the Jiajia tea.
That's right. Specific material from food residuals in the mouth favor the division of certain microbes. For example, high sugar intake reduces richness and diversity, while increasing Streptococcus and Rhodella abundance in mouth swab samples. Oral microbiome composition also differs between hunters with high meat diets and crop farmers who mainly eat rice and vegetables.
So, though it helps to restrict subjects from eating some foods known to alter the oral microbiome, we can't really control their overall nutrients intake, which may vary over the washout and intervention periods.
Any ideas on how to deal with this?
Maybe we can track their dietary intake using a 24-hour dietary recall. Results can then be adjusted statistically by adding nutrient intake as covariates.
Another limitation is our small sample size of 6 pairs. It is difficult to generalize findings to the larger population as the samples may not represent its diversity.
Yep. Outliers may also disproportionately influence your conclusions due to higher variability. It would be here to gather more subjects in your future studies.
Got
Okay,
it.
thanks for